Good afternoon, everyone. I would like to welcome you to today's webinar titled Genetic Research in Eating Disorders, a Primer for Clinicians. Before we begin, there are a few things that I'd like to note. Participants will be muted upon entry and videos turned off. For technical assistance, we ask that you use the chat box located at the bottom of your screens. You will also receive an email in approximately 3 months requesting feedback and impact on today's presentation. Lastly, we ask that you visit our website at www.ncdus.org/training to view other training opportunities that we may have. At the end of this webinar, you will also have an opportunity to ask any questions that you may have. Any unanswered questions will be sent within 1 week from today by the presenter. I'll now like to go ahead and introduce today's presenter. Today's presenter is Dr. Cynthia M. Bulick, who is the founding director of the University of North Carolina Center of Excellence for Eating Disorders, a distinguished professor of eating disorders at the University of North Carolina at Chapel Hill, and professor of nutrition in the Gillings School of Global Public Health. She is also a renowned clinical psychologist. She's also professor of medical epidemiology and biostatistics and director of the Center for Eating Disorders Innovation at Karolinska Institute in Stockholm, Sweden. Dr. Bulick received her BA from the University of Notre Dame and her master's and PhD from the University of California, Berkeley. She completed internships and postdoctoral fellowships at the Western Psychiatric Institute and Clinic in Pittsburgh, Pennsylvania. Dr. Bulick has developed eating disorders programs in New Zealand, the United States, and Sweden, and has active collaborations all over the world. She has published more than 640 papers and 50 chapters on eating disorders. She's author of 7 books, including Crave, Why You Binge Eat and How to Stop, The Woman in the Mirror, Midlife Eating Disorders, Your Journey to Recovery, and Binge Control, a compact recovery guide. Dr. Bulick has been the recipient of numerous awards, including the Eating Disorders Coalition Research Award, the Academy for Eating Disorders Leadership Awards for Research and Advocacy, the Price Family National Eating Disorders Association Research Award, and a Don and Melissa Nielsen Lifetime Achievement Award from the National Eating Disorders Association. She's passionate about advancing the science of eating disorders and translating science for the public. I'll now turn things over to today's presenter, Dr. Cynthia Bulick.
Thanks so much, Lachelle, for that introduction, and welcome everyone to Genetic Research in Eating Disorders, a Primer for Clinicians. Disclosures: Shire Pharmaceuticals, Idorsia, Pearson, and Equip Health, Inc. And my gratitude quilt. The research you'll hear about has been funded by these various groups. And on to eating disorders. I think we missed a slide. So the talk map is we're going to start talking about why I even studied the genetics of eating disorders, give you a basic understanding of current results, and then really try to get into guidelines for clinicians about how you can use this information today in your practice, and then talk a little bit about EDGI, which is our current study, the Eating Disorders Genetics Initiative. So just the basic topography, I think pretty much everyone here knows the primary eating disorders are considered to be anorexia nervosa, bulimia nervosa, binge eating disorder, and ARFID. And the epidemiology, as far as we know, is anorexia between 1% and 4%, bulimia 1% and 2%, binge eating disorder 1% and 4%. ARFID is still a question. We don't have real strong epidemiology on it yet. And we know that all of these disorders have increased mortality. We also know from decades of twin research that these disorders are heritable. Anorexia was about 60%, bulimia about 60%, and binge eating disorder about 45%. And those numbers came from twin studies and basically talked about the percentage of liability to the illness that is influenced by genetic factors. But today we're going to go one step further, and we're going to start talking about genome-wide association studies. So not just what the twin studies told us, but how we're actually going in there and starting to identify the genes that influence risk for these illnesses. And of course, how they interact with environmental factors. So real importantly, and you probably all see this in your practice, is we know that eating disorders tend to fluctuate or migrate across clinical presentations during the course of someone's illness. And this is from the Swedish quality registers at baseline when people are first registered, at 1 year follow-up, and then if there's another year follow-up included as well. And just shows you all these little lines, the frequency and the extent to which these disorders do not breed true. And when we are studying someone in our office, we're really getting a snapshot of their illness at that point in time. Why do we do this? Why do we even study the genetics of eating disorders? From my perspective, as far as I'm concerned, eating disorders defy biology, and that's one of the things that makes them incredibly interesting. For people with anorexia, starvation is reinforcing. They feel better when they're starved. Hunger cues are dysfunctional in all of the eating disorders in one direction or another or both. Bodies, especially in anorexia nervosa, seem to revert to a negative settling point. And I'm not saying set point on purpose. I'm saying that even after we renourish people with anorexia, their bodies and their minds tend to pull them down to a previously low weight. We know that there's this perplexing hypermetabolic period when we renourish people with anorexia nervosa, when their body is burning through calories and it requires so much food to help them gain or maintain weight. People with anorexia say that fats are aversive. The rest of the world tends to really like fats, but they don't like the feel, the mouth feel, the taste, the sensation that it gives them when they eat fats. Satiety cues are overridden. So that stop sign that you have when you're too full either doesn't exist or gets eroded over time in people who have binge type eating disorders. And activity is more reinforcing than food for people with some forms of anorexia nervosa. And we have animal models of that at this point where we know that even in the presence of their most delicious favorite food, mice can be conditioned to run themselves to death. We need to rescue them before they get to that point. And I also often talk about the negative energy balance trap. We don't know what causes this, but people with anorexia nervosa prefer being in negative energy balance. And that's when the amount of energy expended through exercise, physical activity, rest, fidgeting, purging is more than the amount of energy consumed as food. And this is one of the things that we need to understand desperately biologically. The other thing is the study of eating disorders genetics doesn't just tell us about eating disorders. It tells us about all of those co-occurring conditions, anxiety disorder, depression. It tells us about the genetics of metabolism, of nutrition, of physical activity, and about obesity. So it really is just a starting point for all sorts of information that can emerge from the study of genes and environment in eating disorders. So here's the basics. I said in the beginning, we're going to talk about GWAS, and I'll just give you a real sort of outline of what that actually means. And I'm skipping over like decades of research in which we just studied one or a few genes. Now what we do is we get very large samples of cases or individuals with an eating disorder, a specific eating disorder, and compare them to very large samples of controls. And those are people with no history of an eating disorder. And they need to be matched on ancestry because you want to make sure that any differences that you find are actually due to the illness and not due to the fact that you might have different ancestries present in your cases and in your controls. Now, instead of just studying one or a few genes, in a GWAS, we take people's DNA either through saliva or blood, and we genotype it, and then we slather millions of markers across the genome. So we're actually making millions of comparisons. And what we're looking for is at each one of those spots on the genome where things differ. So here you can see our cases have Cs, our controls have Ts, and that's a locus or an area on the genome where they differ. And that's basically what those markers do, is they look across the entire genome to see where the differences between cases and controls lie. The output of the GWAS, and if you've heard me talk before, you know that the reason it's called a Manhattan plot is because if you're lucky and if you identify a lot of loci, these towers start looking like the Manhattan skyline. And this is actually from an early schizophrenia GWAS. And what you see is on the bottom, you see the human chromosomes, including X way up here. And on this axis, you see the significance level. And remember, I said we were slathering 1 million markers across the genome. So that also means we're doing 1 million statistical tests. So if you think back to your statistics classes, you know the more comparisons you make, the more likely you are to get a false positive. So we have to correct for doing 1 million comparisons. So we set a very stringent p-value in order to say, yep, one of these markers is actually significantly different between cases and controls. And the reason that's important is not just scientifically, but once we identify a locus and identify the genes in that locus that might be operative, we start investing a lot of money into exploring exactly what that gene does. So we don't want to be wasting money and our neuroscience colleagues' time on false leads. We want to make sure that we're confident that that really is a causal gene. And a lot of the work that you're going to talk about today is done by the Psychiatric Genomics Consortium. The PGC is the largest experiment in the history of psychiatry. We have over 800 clinicians and researchers from around the world studying many different psychiatric disorders. I am the founder, I guess, of the PGC-ED, the Eating Disorders Working Group of the Psychiatric Genomics Consortium. And this group meets whenever we can in person and frequently via Zoom, more recently, in order to keep our science going. Now, the main, I will say, leap forward in genetic research for anorexia nervosa was done by the Anorexia Nervosa Genetics Initiative, which was funded by the Klarman Family Foundation. And that took part, it was centered at the University of North Carolina at Chapel Hill, where I am. And then we had other groups at Karolinska Institute in Sweden, QIMR Berghofer in Australia, with help from the University of Otago in New Zealand, and Aarhus University in Denmark. And what that freeze did, so that a freeze is when you basically say, no more data, we're going to analyze these data now. We had 33 data sets from 17 different countries represented by all of these flags, and almost 17,000 cases and 55,000 controls. And as you can see, if you look at the composition of that sample that came out of freeze 2, 74% of it was due to the effort of the Anorexia Nervosa Genetics Initiative, or ANGI. And these are the results. And I'd like to just tag Hannah Watson here, who was the first author on this paper that came out in Nature Genetics. And what you can see here, since you know how to read a Manhattan plot, is that our Manhattan plot indeed showed 8 loci that were above that critical line of significance. Not as many as schizophrenia, where you saw 108, but this is a good start, given that our sample size, 17,000, was actually still pretty small. And what you can see here is that the loci that were significant had been implicated in other GWASs that were done on various autoimmune, metabolic, neuropsychiatric, and sex hormones. So some of these loci were implicated in obesity, some of them in various autoimmune illnesses like IBD and ulcerative colitis and Crohn's, and again, some more obesity-related traits and BMI. And you have to be a little bit careful. I think that tendency when you look at your GWAS results is to tell a story right away. But this is actually very early stages. We anticipate that we're going to have hundreds of significant loci, much like you saw in schizophrenia, as our sample size grows. But that's not all you can get out of a GWAS. We talked about that information above the red line. There's also a lot of information below the red line. And the way to think about it is that as you increase your sample size, a lot of those loci are going to start sort of like rising up and becoming more significant as your sample size grows. But there's still information below that red line that you can tap before they become significant. And we're going to talk a little bit about genetic correlations. And what these are, and I think it's super important to make sure you're separating out what we typically think about as correlations, and that is For example, if you look in the clinic, anorexia nervosa and obsessive compulsive disorder are correlated on the phenotypic side, or the part that we actually see. But that has nothing to do with genetics. What we're talking about with genetic correlations is that you're actually looking at the same genes influencing the trait. And what genetic correlations do is we can take a sample of someone with, or a group of people with anorexia in the United States, and we can compare them with someone in Germany who had IBD. We have 2 large samples, we have 2 GWASs, and we can use the summary statistics that come from their GWAS to see whether there are any genetic correlations between those 2 traits. And this is a major advance because in the past, we would have to have those 2 traits measured on the same group of people. But analytically now, we don't have to do that. We can actually look at what is functionally between diseases, the genetic analog of comorbidity. And this approach was actually developed by my kid, by Brendan Bulick-Sullivan, who you see here on the bottom. Now, we're going to start here with this big picture, and then I'm going to pull it apart. What you see here is basically the genetic topography of anorexia nervosa. The way you read the genetic correlations is this red line that goes up and down the middle is 0. So that means no correlation. Everything to the right is a positive genetic correlation. So that means the same genes influence the traits in the same directions. And I'll unpack this as we go through. And on the other side, on the left side, are negative genetic correlations. So you have the same genes influencing them, but in opposite directions. And I'm going to go through section by section. We're going to start at the very top with the things in green. So everything we're talking about on this slide is a positive genetic correlation, the same genes acting in the same direction. And just like I talked about with the phenotypic correlations, the strongest genetic correlation we saw, and in fact, one of the strongest genetic correlations in psychiatry, period, is between anorexia nervosa and obsessive compulsive disorder. So same genes, increasing risk for both disorders. We also see positive genetic correlations with other psychiatric traits, major depressive disorder, schizophrenia, anxiety, depressive symptoms, and neuroticism. And then right below that in lighter green, we see positive genetic correlations with various measures of educational attainment. And this is really interesting and might lead you to rewriting your book about high educational achievement in anorexia nervosa, because there have been decades of information about high achieving families and putting pressure on their kids to succeed. But these data suggest that there's actually a genetic correlation there. So some of that high achievement orientedness might actually be driven by the same genes that influence risk for developing anorexia nervosa. And the next little circle really made me rewrite my book about physical activity in anorexia nervosa. So there's a positive genetic correlation between anorexia and measured physical activity. And again, what that means is the same genes that influence risk for anorexia nervosa are also associated with high physical activity levels. So some of the challenges that we face in getting people to reduce their physical activity as they're recovering from anorexia nervosa is actually sort of fighting an uphill battle against their biology, because they might be genetically predisposed to be highly active people. The next set, as you'll see, remember that red line up and down the middle, everything except high density lipoprotein is over here on the left, or a negative genetic correlation. So that first part that we saw before with all the green dots makes perfect sense. Anorexia rests firmly in a cluster with other psychiatric disorders. But less expected was the fact that we see these negative genetic correlations between anorexia nervosa and metabolic factors like insulin resistance, fasting insulin, leptin, type 2 diabetes. So the genes that increase your likelihood of developing anorexia nervosa are associated with lower likelihood of developing type 2 diabetes or having high leptin, with one exception. And that exception is the one favorable metabolic parameter, which is high density lipoprotein. So your increased genetic risk for anorexia nervosa is also associated with increased risk for that favorable metabolic parameter. And just reminding you again, because I think just because we've been doing this for so long, we tend to go back in our minds to thinking about regular phenotypic correlations. Remember, we're still talking on the genetic level. These are genetic correlations. And then the last piece, again, made me rewrite my book on anorexia nervosa. And basically, these are all negative genetic correlations with body fat percentage, fat mass, weight to hip ratio, waist circumference, fat-free mass, all of these anthropometric measures. So if you have high genetic liability for developing anorexia nervosa, you also have lower genetic liability for becoming obese or gaining weight or having a large hip circumference. So we actually see negative significant genetic correlations with anthropometric measures. I'm going to throw them all together on one slide because this sort of encapsulates what the genetic underpinnings of anorexia nervosa are. And what that means is there's this high cluster of genetic factors that influence risk for psychiatric disorders, especially OCD. But we also see equally strong negative genetic correlations with metabolic and anthropometric traits. So if anorexia was only psychiatric in origin, we wouldn't see these bottom two ovals. We would only see this top two ovals. The fact that we see all of these ovals on this global chart suggests that anorexia nervosa has both psychiatric and metabolic / anthropometric components. So what does this do? How does this help us understand anorexia? It is perplexing to understand how people with anorexia nervosa reach and maintain such a low BMI. As I said in the very beginning, it defies biology. Second, I talked about that frequent return to a negative settling point. Whether something happens metabolically or if there's a predisposition somehow to once someone gets down to that low weight, their body pulls them back down. Again, this encourages us to look at metabolic contributions to that. Negative correlations with BMI and other unfavorable metabolic parameters, and the positive genetic correlations with HDL. So sort of opening up a whole new chapter on what we need to look at in terms of causes for anorexia nervosa. And trying to understand this paradoxical reaction to negative energy balance. Why, when most of the world prefers or finds it more comfortable to be in positive or neutral energy balance, is negative energy balance so reinforcing for people predisposed to anorexia nervosa? Implications. So we went so far, given the strength of those findings, on a fairly small sample GWAS, that anorexia nervosa may best be conceptualized as a metabopsychiatric disorder. We suggested that greater attention to metabolic factors may improve outcome. As we have all encountered, renourishing someone with anorexia nervosa and helping them to actually maintain their renourished weight is a very challenging clinical undertaking. It might provide an explanation for why adequate refeeding is essential for preventing relapse. When we see patients being denied further care when they reach 80% of their ideal body weight, it's almost a guarantee, and perhaps it's a metabolic guarantee, that their weight is going to plummet again. And hopefully this is going to give us data at some point to understand why refeeding to adequate weights is so important. Might also give us a little bit of insight into why FBT is effective in a lot of youth with anorexia nervosa. And that is the emphasis is actually on metabolism, even though it's not directed toward metabolism. That active refeeding, that active making sure that you get someone up to a reasonable weight before you back off is something that can really lead to lasting change. And we think about it psychologically, but there's probably something going on metabolically as well. And I said it before, I'll say it again. The next step is to really dig into what the metabolic mechanisms are. We're going to need more data. We're going to need a larger GWAS to really see where the different loci are and what the genes are that are implicated. But clearly, this is shining the light in the correct direction. Now, I've been talking about anorexia up till now, but as with every other aspect of eating disorders, one size does not fit all in the genetics of eating disorders. But since we haven't done genome-wide association studies for bulimia, binge eating disorder, or ARFID yet, we needed to go about this in a different way. And we're going to talk about polygenic risk scores. This is another technique that looks below that red line and looks at all of the information that you have available, as well as above the red line, all of the information that you have available on a particular disorder or disease. And what you do is you take a sample on which you've done a genome-wide association study, like the one that you just saw, and you create a single score. It is a single score of an individual's polygenic load. How many of those risk alleles do they have? How high is their genetic risk for anorexia nervosa? You can then take that calculation and you can apply it to a second sample or an individual and basically ask for a whole bunch of people who weren't analyzed in that first sample, what is their polygenic risk for anorexia nervosa? And we'll be clear, this is not a genetic test. This is just based on what you know today, how strong your GWAS is, what an individual's polygenic risk is. The future of what we can do with polygenic risk, and this paper down here, if you type in the PubMed ID 330-52393, that'll take you straight to a really good paper about polygenic risk scores. In the future, they may help us improve risk assessment by combining polygenic risk scores with other measures of risk. They may be able to help us screen for mental health disorders in the population. They may help us in making a diagnosis or clinical decisions. When someone walks into your office for the first time today, showing signs of anorexia nervosa, you don't know whether they're at increased risk of developing a severe and enduring case. That's something that PRS might help us be able to distinguish as they become more refined. Likewise, are certain psychiatric PRSs associated with treatment response? Will we ultimately be able to say, based on your polygenic risk scores and perhaps some other indices, you might respond better to this treatment than to that treatment. And that would keep us from wasting time with that treatment if we know on a biological level that the person is more likely to respond to the other treatment. And one more time. Are PRS associated with adverse physical health outcomes in mental illness? And one of the ones that I'm very interested in is whether we'd be able to predict on the basis of polygenic risk scores who is at risk for developing extreme weight gain and antipsychotic medication. Not so much in anorexia populations, but in people with schizophrenia and bipolar disorder. So here's what we did. To say, are there differences on the genetic level between anorexia, bulimia, and binge eating disorder, we looked to the UK Biobank in order to apply polygenic risk scores. And I'm going to go through this a little bit quickly because I want to spend as much time as possible on what you use in the clinic. These are the number of people they had: 768 anorexia, 423 bulimia, 561 binge eating disorder. And we see another type of topography, like I showed before, with the genetic correlations. But here, we're looking at odds ratios of different disorders. And we're going to start again with the psychiatric disorders. This time, everything to the right is a positive odds ratio. Everything to the left is a negative odds ratio. And the filled in dots are the significant difference. But really, we're going to be looking at the direction of effect here. And what I want you to see in this, pretty much, we're seeing some of the same patterns across the 3 eating disorders, that they all tend to fall to the right of that 1.0, which is the center line, meaning that we have positive odds ratios for all of the psychiatric disorders. Again, suggesting that anorexia, bulimia, and binge eating disorder are firmly anchored in the psychiatric realm with some differences. Then when we go a little further and we look at the metabolic realm, we see different patterns. And I think the one that stands out the most is the binge eating disorder, which is the dark purple circles. And even though they're not significant, it's the pattern that is different. So we see elevated odds ratio for type 2 diabetes, reduced for high density lipoprotein cholesterol, and then elevated for fasting insulin. So again, what we had seen in the genetic correlations was basically the opposite pattern in anorexia nervosa. And that also showed up here. So where you see some decrease on fasting insulin. And then this is where we saw the most separation. And everything in the light blue is anorexia nervosa to the left. So reduced odds ratio, totally consistent with what we saw in the genetic correlations. But the difference comes especially with binge eating disorder, where we really see stronger associations with overweight, with obesity, with childhood obesity, with BMI. So I guess the take-home message from this study, without going into the nitty-gritty, is that anorexia, bulimia, and binge eating disorder have clear genetic parallels when it comes to their relationships with other psychiatric disorders. But where they separate is in their underlying genetic relationships with metabolic and anthropometric or body measurement traits, really giving us the first glimpse of how these three disorders might differ on a genetic level. So one size does not fit all in eating disorders. ANBN and BED are broadly similarly associated with polygenic risk scores for psychiatric traits. Exceptions, we didn't go into the details, but there were some difference with ADHD and autism spectrum disorders. ADHD was only significantly associated with binge eating disorder. The differences lie in those polygenic risk scores for metabolic and anthropometric traits. And might we be able to use polygenic risk scores in risk models to predict diagnostic crossover and outcome? Remember that first bubble plot that I showed you? If we knew polygenic risk scores at day one when people were registered, would we be able to say, yep, this person is likely to remit, this person is likely to continue to have anorexia, this person is likely to cross over to bulimia nervosa, and then tailor our interventions accordingly? Why? Ultimately, what we want to do is we want to create an empirical classification system based on genetics. Right now, the DSM-5, the ICD-11 to be are not really based on underlying genetics of these illnesses. But our approaches very much can help us figure out what belongs where. Excuse me. It may help us predict the course of illness and tailor treatment accordingly. It also might help us properly position atypical anorexia relative to other eating disorders. So atypical anorexia, in a simplified way of explaining it, is having all of the symptoms of anorexia nervosa except not being at low weight, although that is an inadequate explanation. And the question is anorexia the same genetic illness at any weight? Or are there actually differences in people who tend to be on the low side of the BMI spectrum? It will help us properly position ARFID subtypes relative to other eating disorders, but also relative to other psychiatric disorders, and ultimately move toward personalized medicine approach to treatment instead of a one-size-fits-all. Also, hopefully, and another one of our goals is drug repositioning or drug development based on the genetic results. We have no medications for any eating disorder that are actually based on the disorder biology. And hopefully that's where we'll be going as our GWASs become stronger. And ultimately, eliminate mortality, and decrease the lives lost from the eating disorders. Now, critical limitation. Much of the work that's been done so far has been done on European ancestry populations. And in part, that's an artifact of where the analytic techniques and the genotyping was a decade ago. That is no longer an issue. We now have the ability to study, using GWAS, trans ancestry populations. And it is critical to have representation from diverse populations to ensure that we don't perpetuate or amplify health disparities. And that also includes making sure that men are involved in our genome-wide association studies. But the goal ultimately is not just to identify the genes, but to fully account for the impact of both genes and environment, and how they act and how they co-act to influence risk for eating disorders. And some of the domains of diversity that are really important to have represented in our GWAS are biological sex at birth, gender identity, weights and shapes, genetic ancestry, culture, and socioeconomic backgrounds. Now, what do you do with this information today in your office? Science communication has become, unfortunately, a political issue. Anti-truth and anti-science are rampant, and the internet doesn't help. It is the scientist's responsibility to interpret and contextualize the information that comes from our science. It's the clinician's responsibility to just have a general understanding and assist patients and families with weaving the science into their own personal narratives of their experience. And there is a very important role for genetic counseling and eating disorders, but as of yet, we still don't have a strong, not much at all actually, of genetic counseling for these disorders. So this is a way, when parents or patients come to us, when groups come to us and say, help us understand, this is a simplistic but helpful, relatable model of how to explain the impact of genes and risk, genes and environment. And I break it down into a card game. I mean, everybody basically knows the 4 suits of a card game. And I say, these are the 4 domains that influence your risk for developing an eating disorder. The spades, for example, are your genetic risk. But something we don't study, because it's actually harder to study, is there are also genes that are protective. And I think it's very important for people to remember that you can have a high genetic predisposition for anorexia nervosa, and maybe that came from one side of your family, but you only get 50% of your DNA from your mom or your dad. And the other 50% might actually have some genes that are protective or that buffer against that genetic risk. So you have to put that into sort of your narrative or your conceptualization of risk as well. And then alongside that, you have environmental risk factors. So anything out there that we know can influence your risk to maybe go on that first diet or to experience your first binge. Things like bullying, things like engaging in sports or activities that demand low body weight, any kind of teasing, any of the environmental risk factors that could influence or maybe even activate your underlying genetic predisposition. And then the 4th group is environmental protective factors. So yes, you might be exposed to some of those environmental risk factors, but they too might be buffered by environmental factors. If you're able to come home and talk to your parents about being teased, and they can sort of help you work through that and develop your defenses, that can act as a protective factor and buffer some of that environmental risk. And in fact, some of the environmental protective factors can also buffer some of the genetic risk. But the other thing is, you can have fairly low genetic risk for an eating disorder, but if you just have environmental risk factors piled on high, that can probably make someone who actually has low genetic risk for an eating disorder develop an eating disorder. But at the core of it, you're dealt a hand, and the black cards, the spades and the clubs, you get them when you're born. Those are the genes that you have with you at the moment of conception, and they stay with you to the very end. And then you pick some cards, and that includes some environmental and some environmental protective factors. At this stage, we can't do anything about the spades and the clubs, so they're sort of fixed. The only thing we can do is influence the environmental risk factors and environmental protective factors. That's what psychotherapy does. That's what we do when we try to make our families healthier places to be. And that's really where we can focus our interventions at this point. Hopefully, from the work that we're doing, we'll be able to perhaps find some druggable targets that we can use to not change the genetic factors, but to influence their expression. Now, the one thing that I'm going to add to this is there are some conditions where if you have a certain gene variant, you're going to get that illness, or you'll have a 50% chance of getting that illness, like in Huntington's. We haven't found any rare variants or genes like that yet that influence risk for eating disorders. So I'm fairly comfortable telling families and patients that it really is the spades and the clubs and the hearts and the diamonds that you need to think about when you develop your own narrative about how genes and environment influence your eating disorder. Now, how can a genetic counselor actually assist with this? Genetic counseling is a really important area in which we need to develop more people who know how to do it for psychiatry, and within that group, more people who can do it for eating disorders. Genetic counseling for mental illness helps alleviate stigma and shame. It can correct misconceptions about the condition. It can prepare family members to intervene if necessary, if there's a high risk of their offspring developing a condition. It definitely promotes help-seeking behaviors. And even just sort of baseline evaluations, because I think there's a little bit, people become a little hypervigilant if they know that there might be an increased risk of their offspring having an illness. But there's never any harm in getting a baseline evaluation and having another professional cast their eyes over your child to see if there appears to be any risk factors emerging. And it promotes help-seeking behaviors. There's only been, I think, as far as I can tell, one study on genetic counseling and eating disorders, and that was done by Julianne Stroikens, who was at UNC Greensboro, who was a genetic counseling student. And she surveyed 107 individuals who had a personal history of an eating disorder. And what she did is she had them estimate what they thought the risk to their children was. And then she also gave them a vignette about a genetic counseling session with someone with an eating disorder, and saw how it influenced their perceptions of an eating disorder. Now, the most important thing, the one result that just really stood out is that every single participant who had an eating disorder overestimated the risk of their children developing an eating disorder. So the empirical data suggested it was 70.5% increased risk. They estimated 43% higher for sons, 57% higher for daughters, way high. And 100% of them overestimated the risk to their daughters. Again, because we all have this sort of internalized perception of eating disorders being more frequent in women. And I think that bias also showed up in these results. And then this is the pre and post imagined experience. So, this vignette basically talked about how it talked them down from their overestimations. It talked about what the actual estimations were, and just gave them a lay but accurate understanding of the role of genes in eating disorder risk to their offspring. And without going into every single line, I think, again, this is a pattern that is incredibly it recurs, and it's incredibly obvious that after that imagined experience, their perceptions all moved toward the healthier direction. So eating disorders were less stigmatized. There was less shame. They were less looked down upon. There was less parental guilt. And they felt like someone with an eating disorder is less likely to be treated differently because they had an illness. And I think this particular one here, the guilt measure, is one that's just so important to me because, I mean, I've been in this field for a long time and I have seen parents be guilted about, or feel guilty or be guilted about their children having eating disorders, you know, from day one when I started in this field. And I think this approach can really help reduce guilt in parents whose children have an eating disorder. And just real briefly, the one thing we don't want to do is we don't want people to have genetic guilt, like to start feeling guilty about passing on their genes, because if there's one thing we have no control over in this world, it's you know, what pops out in the sperm and the egg, and which genes get assorted and turn into a person. And this is just a little commentary from one of the participants. Wow, just reading this has validated so many things. I felt like that, like it's the genes and environment, and that guilt is so common in people with eating disorders. I feel like people need to hear and understand this, both people with the eating disorders, and the public. And I agree with her on that one. And then our responsibilities. So, you know, with my scientist hat on, it's my responsibility and our responsibility not just to do the science, but to package the information for patients and families in a digestible way. With my clinician hat on, it's our responsibility to make an effort to integrate genetics into our own case conceptualizations and develop comfort with answering our patients' questions. And, you know, I think we do, to some extent, always ask if there's someone in the family who's had an eating disorder or OCD, depression, anxiety disorders, but also thinking about those spades and clubs and hearts and diamonds, and the relative contribution of each. I think is sort of a useful way to start assorting in your own mind what the different factors are that have influenced the person's development of an eating disorder who's sitting in your office. We have to know our limits. If we live in a country with genetic counseling like the United States, make use of your colleagues. And if not, find whatever resources you can for patients. You know, there are family groups all around the world. Feast is one of them that you can just connect with and find other parents who have gone through what you're going through. And just like everything else, we must never perpetuate misinformation about eating disorders. And I think that's really hard for us. And it's hard because it is much more difficult to get people to unlearn things than it is to get people to learn things. And we have to get to the point where we're practically bombarding people with accurate information in order to erase all the misconceptions that have developed about these disorders over the course of decades. Here's some messaging do's and don'ts, just to help out. First off, genetics is just one piece of the risk puzzle. All or nothing thinking, out the window. So whenever you hear someone say genes or environment or nature or nurture, just stop them and gently have them replace those ors with ands. It's hard for some people to understand probabilities. And that really is what genetics is all about. And again, that's why I use those 4 cards, because I think it really helps people put probabilities in their head rather than all or nothing thinking. As I mentioned, steer people away from genetic destiny. There's no such thing as genetic destiny in eating disorders. And again, genetic guilt. We have no control over what comes out. And also, we're not saying that eating disorders are genetic and not psychological. They are both. We absolutely will always still have to do lots of psychological work in the treatment of eating disorders. But hopefully we'll be able to bolster that by intervening on the biological level as well. and genetic simplification. If you ever see a headline that says something like, I found the gene for eating disorders, they're lying. There will never just be one gene. There will be hundreds of genes that influence risk for eating disorders. And as mentioned before, also a lot of buffering genes that might counteract some of those effects. And I can't see my last point. So whatever the last point is, I suggest you take a look at it. Now, this is just one little quick slide. And this is why polygenic risk scores are not a genetic test. What these two curves show you are the polygenic risk scores in red of people who have a disease, and in blue, the polygenic risk scores of people who do not have that disease. And maybe out here, we might find some groups of people who only have the disease, but as you can see, there's a lot of overlap. So just saying someone has a high polygenic risk score, all these people here might have a high polygenic risk score, but they don't have the disease. So in no way does this approximate a genetic test. And if anyone tries to sell you a genetic test for anorexia nervosa, please don't waste your money, because that's not where we're going. And there is no genetic test for anorexia nervosa or any other eating disorder. And then just in my last minute, we have now transitioned from Angie to EDGY. We have funding from the National Institute of Mental Health to broaden Angie beyond anorexia. And we're now including anorexia, bulimia, and binge eating disorder. We're hoping to be able to add ARFID. And in some of the other countries where EDGY is operative, we're also including people with atypical anorexia nervosa. So hopefully we're eventually going to have the whole DSM represented in EDGY. Our goal is 100,000 people with eating disorders plus controls. We're diversifying our samples to make sure that we actually reach those goals. Everything is online. So we now do digital consent and questionnaires and simple at-home COVID-safe saliva collection. In the past, when we had to draw blood, these were pretty hard studies to do, but the field just advances so fast that saliva works just as well now. We're engaging the advocacy community, and we're engaging clinicians. And we are not just looking at genes, and we are not just looking at eating disorders. So our online phenotyping, the questionnaires cover life events, trauma, physical activity, OCD, alcohol and drug use, tobacco use, anxiety, depression, and their treatment history. So we're getting a full picture of everyone who participates in EDGY. So we really can look at how genes and environment act and co-act. This is our website. It's edgy.org. We have it in English and in Spanish in the US. And we've globalized. So NIMH is funding US, New Zealand, Australia, and Denmark. National Institute of Health Research is funding EDGY UK. The Swedish Research Council is funding EDGY Sweden. We're about to start EDGY Mexico with Eva Trujillo in Monterrey. We have a small grant to start EDGY Taiwan, and we're working on Italy, the Netherlands, and Canada. So, and any other countries out there, give me a call, drop me an email, because we really want to globalize this study. And these are the people that keep it running, for whom I'm deeply grateful, everyone on the UNC side, as well as the whole eating disorders working group of the Psychiatric Genomics Consortium. And that's us. Follow us on Twitter, go up to the website, take a look at EDGY. Anything you'd like to do, if you have any questions, drop me an email or tweet me. Again, thank you so much. And now it's time for questions. Sorry, go ahead, Lucia.
No, that's great. Thank you so much, Dr. Bulik, for a wonderful presentation. We are now going to begin our question and answer segment. I just wanted to remind you all that you will receive the handout from today's presentation along with the evaluation for you to fill out, and that will be sent immediately after the webinar. Our first question says, thank you for a fantastic presentation. How do you deal analytically in these genetic studies with the fact that many individuals have lifetime diagnoses of more than one ED diagnostic crossover? How are such individuals binned if you are trying to differentiate genetic signatures of BN from AN, for example?
Excellent question. Yes, so we do it two different ways. One way that we do it is a hierarchy way. With AN, if someone has had AN at any time in their life, they get put into the AN bin, to use your terms. And then the same thing, if a person has had BN and BED, then they get put into the BN bin. But then we also sort of rearrange it in other ways so they're not hierarchical. And we can include anybody who's ever had anorexia, anybody who's ever had bulimia, anyone who's ever had BED in separate bins, knowing that there might be some representation of each person in more than one bin. But for most intents and purposes, we use that diagnostic hierarchy where, I mean, especially with AN, people who had AN at any time in their life representing the core group.
Thank you so much, Dr. Bulick, for your response. The next question reads, are there individuals whose eating disorders are more influenced by genetics than environmental factors and vice versa? In other words, do all individuals with AN get influenced by these genetic markers low-key in the same way?
Oh, I love these questions. That's another great question. David Clinton and Andreas Birgergård in Sweden have a study going on. If you remember that bell curve I showed you of the polygenic risk scores, they have selected people with anorexia nervosa, and we're starting with anorexia because that's what we have the GWAS on, who are in the top decile of the polygenic risk scores, and from the bottom decile of polygenic risk scores. So those people have both developed the illness at some point in their life, but their underlying genetic risk is very different. One being the highest possible bin, one being the lowest possible bin. And they're doing intensive interviews and a qualitative approach to figure out what's different. How does their eating disorder differ? How do their environmental triggers differ? Because theoretically, what we imagine is that someone with high polygenic risk doesn't need a lot of environmental perturbation to push them over the hill and develop the illness. Whereas someone with low genetic risk might actually need a whole bunch of environmental factors to push them over the hill. But that's just theory. But now that we have genotypes, we can actually look at that. I do not have an answer for you, but I do have a promise that that study is underway. And once we get some results and get it published, I'm sure we'll do a blog post on it and let people know what we found.
Thank you so much for that response, Dr. Bulick. We have one more question here. To identify genetic risk factors to eating disorders more broadly in case conceptualization, would it be helpful to include questions about those metabolic markers? Do you have relatives who had struggled with insulin resistance, not just psychiatric family history?
Another good question. Yes. In fact, that is a direction that we definitely need to go in. One of the things that we can do in Sweden and in other Nordic countries is connect our genetic data with the national patient registries. So we would actually be able to, without even asking the person that question, because we don't always know about all of our family's medical illnesses, we would be able to connect to their family's medical data and see if there is increased risk of some, or increased presence actually, of some of those traits in the medical records, in the diagnoses. So that's an excellent idea. And let me know who you are, so that I can attribute that idea to you when we do that research, because it's a great idea, because the expectation is that we would see differences in the family members of people who had different eating disorder diagnoses. So I think that's a very testable hypothesis and one for which I am grateful.
Thank you once again, Dr. Bulik, and we will have enough time to address our last question. That question reads, I've never heard of genetic counseling. I am very interested. How did you get into this field and what did you master in?
Yeah, well, I'm not a genetic counselor. I kind of wish I did have a side degree in that. It is a wonderful field, and it really helps people determine how much at risk their children are. You know, for some diseases that are strongly genetic, where your child might have a 50% chance of developing the illness, you know, they do a great job talking you through that with you, with your partner, making decisions about reproduction. And for other disorders where there's a much more gray area like anorexia, you know, obviously in the study that Julianne did, people grossly overestimated the risk to their kids. And we have patients come to us all the time who are saying, I don't know if I want to have kids because I don't want my child to go through what I went through. But that's the assumption that there's like 100% risk, which is way off base. So I think it really does, it helps people put things in context, give them some real numbers, understand probability, and helps them plan what their reproductive life is going to be like. And there is a subset of people who are genetic counselors who are psychiatric genetic counselors. We just don't have a lot of them yet who specialize in eating disorders.
Thank you once again, Dr. Bulik. That is amazing. We truly, truly appreciate everyone attending today's presentation. We have addressed all of the questions. We want you to definitely be on the lookout for the evaluation and the handout from today's presentation. And Dr. Bulik, if you have any last words, we can go ahead and adjourn.
Just everybody go up and take a look at edgy.org and spread the word. We want to get as many people involved as possible so we can get some great answers and improve treatment for all eating disorders. And thanks for your time.
Thank you as well. And thank you all for attending today's webinar presentation.